welcome to all of you present over here. Thank you to all distinguished guests to join us and give us some great time out of your busy schedule. We are pleased to welcome all of you, and first of all I would like to express my gratitude to the chairperson of this program, chief guest, faculty, science faculty members, junior brothers and sisters, and all the entire team of Chiton Biological Research Center. And Chiton Biological Research Center has been recently established by us with 7 guys of BSc 4th year and 2 members of our Balakumari College, Mr. Anil Shetty sir and Audumoni Bhattacharya sir. We have recently established this organization, and this is our first program supported to complete this World DNA Day talk program. So once again I would like to heartily welcome all of you, and I hope you will support us till the end of the program to make it successful. Thank you.
Welcome to each and everyone present here. In this program entitled our Talk Program on DNA, organized by Faculty of Science and supported by Chiton Biological Research Center and Lions Club of Balakumari College. For supporting, we'd like to thank all of the organization. Myself, Sapna Bhatrai, your MC for today's program. And I would like to thank all the people who are present here for supporting such a great programs, such a great program. So, without delay, can I start the program? Yeah. Now, without delay, I would like to.
Because it will encourage us for the better life, towards knowledge, acquiring knowledge, towards achieving knowledge, and towards communicating knowledge about DNA. Because the slogan of the day, World DNA Day, is learn more about it. It is the slogan, or what, sir? You kids have slogan. What is this? Kids have slogan. My reflection. Okay. Learn more about DNA. I am a little bit student of science. I was student of science under up to bachelor's level, but my major subject was physics. I have a little 0.1% knowledge only about on DNA, but I have my जति मैले जानेको छु म के कुरा भनौँ, जति मैले बुझेको छु कि DNA भनेको पहिले यस्तो साइन्स हो नि, यो साइन्स क्रिमिनोलोजीमा पनि प्रयोग भएको छ। यो साइन्स संसारको कुनै पनि अन्तर्राष्ट्रियमा बस्ने र चाहिँ कुनै अर्को ग्रहमा बस्ने जीवहरू केही छन् भन्नेमा पनि अध्ययन भएको छ, र कतिपय कुरा त फोसिल स्टडीमा पनि यबाट अध्ययन भएको छ। त्यसकारण यसको एरिया, यसको यति धेरै ठुलो भएर गएको छ कि तपाईँ हामीलाई यहाँभित्र बस्न, यो DNA भित्र रम्न, DNA भित्र नै हामी डुब्नको लागि पर्याप्त अवस्था छ। त्यसकारण आजको दिनले लाग्छ तपाईँ सम्पूर्ण भाइ बहिनीहरूलाई DNA मय होस्, DNA मा केही कुराहरू तपाईँले दिन सक्नुहोस्, यो दिनले प्रेरणा दिनुहोस्। आजको दिनबाट म DNA मा केही न केही कुरा गर्छु, देखाउँछु भन्ने कुरा प्रेरणा मिलोस् भन्दै यो कलेजको प्रोग्राम जसले सपोर्ट गर्नुभएको छ, सपोर्ट बाई चितवन बायोलोजी रिसर्च सेन्टर, लायन्स क्लब बालकुमारी कलेज, उहाँहरूलाई पनि विशेष प्राध्यापक सङ्घको सभापतिको तर्फबाट उहाँहरूप्रति हार्दिक आभार गर्दै सबैभन्दा मुख्य आभार त बालकुमारी कलेज फ्याकल्टी अफ साइन्स उहाँहरूलाई व्यक्त गर्न चाहन्छु। यसको लिडरसिप गर्नुभएको अनुसार सपोर्टिङ गरेर एक्टिभली पार्टिसिपेट गरेका विद्यार्थी भाइ बहिनीहरू तपाईँहरू सबैलाई म एकदमै मेरो व्यक्तिगत तर्फबाट, प्राथमिक सङ्घको तर्फबाट एकदम warm wishes and warm congratulations to all of you by raising my hand. Thank you.
That's what we know from each other, right? Don't listen to me. Excuse me.
प्रमुख अतिथिज्यू, विशिष्ट अतिथि र अन्य अतिथिज्यूहरू, उपस्थित सम्पूर्ण प्राध्यापक साथीहरू र विद्यार्थी भाइ बहिनीहरू, सबैभन्दा पहिला बालकुमारी कलेजको साइन्स फ्याकल्टीले आजको यो अन्तर्राष्ट्रिय डिएनए डेलाई जुन अत्यन्त महत्त्वपूर्णका साथ लिएर जुन यो कार्यक्रम गर्नुभयो र यो कार्यक्रमबाट हामी सबैलाई जुन एउटा अझ बढी सचेत बनाउने अवस्था निर्माण गर्नुभयो यो फ्याकल्टीलाई म धेरै धेरै धन्यवाद दिन चाहन्छु। र सँगसँगै डिएनएको बारेमा म साइन्सको विद्यार्थी पनि होइन, म म्यानेजमेन्टभित्र पनि इकोनोमिक्सको विद्यार्थी हुँ, जो कारणले गर्दा यो बारेमा मलाई त्यति धेरै नलेज छैन। तर पनि जुन नलेज तपाईँहरूले प्राप्त गर्नुभएको छ र शिक्षकहरूबाट सिक्ने काम विद्यार्थीहरूले गर्नुभएको छ, त्यो सिकाइलाई तपाईँहरूले आफ्नो जीवन उपयोगी बनाउनुहोस् र त्यो सिकाइबाट अझ अरूलाई धेरै विषयवस्तुहरू सिकाएर यो डिएनएबाट मानिसको जीवनमा यस्तो यस्तो प्रभाव पार्न सक्छ र भोलिका सन्तानहरूलाई पनि यस्तो प्रभाव पर्न सक्छ भन्ने कुराको जुन एउटा सचेत र जानकारी तपाईँहरूले आजको दिनमा गराउनुभयो भने साँच्चै नै आज हामीले जुन कार्यक्रम गरेका छौँ यो कार्यक्रमको पूर्ण सार्थकता भएको भन्ने मैले ठान्छु। र विशेष गरी तपाईँहरू बालकुमारी कलेजको साइन्स फ्याकल्टीको विद्यार्थीहरू हुनुहुन्छ, तपाईँहरूको धेरै समय क्लासको अध्ययन अध्यापनमा र प्र्याक्टिकलमा बित्छ। हुन त प्र्याक्टिकल भनेको व्यवहारिक पक्ष हो, त्यही व्यवहारिक पक्षले डिएनए सम्बन्धी जुन तपाईँहरूले जानकारी प्राप्त गर्नुहुन्छ त्यसले तपाईँहरूलाई अझ बढी अध्ययनशील र अझ बढी चिन्तनशील बनाउने छ र यो नलेज प्राप्तिले भोलिको भविष्य तपाईँहरूको अझ बढी सुखमय हुनेछ भन्ने कुरा मैले विश्वास लिँदै म अन्तमा बालकुमारी कलेज प्राध्यापकबाट सञ्चालक समितिमा प्रतिनिधित्व गर्ने व्यक्तिको हैसियतले यो कार्यक्रमको पूर्ण सफलताका साथ यो कार्यक्रममा सहयोग गर्नुहुने सबैलाई धन्यवाद। र म फेरि पनि बालकुमारी कलेजको जुन हाम्रो विज्ञान फ्याकल्टी छ र अहिले अनुपमणि बज्राचार्य सरले लिड गरिराख्नु भएको छ, यसमा अत्यन्त सहयोगी साथीहरू हुनुहुन्छ र अत्यन्त एक्टिभ र इनर्जेटिक विद्यार्थी भाइ बहिनीहरू हुनुहुन्छ। तपाईँहरू सबैको अध्ययन र चिन्तनले आजको डिएनए डे मात्रै नभइकन समग्र यो फ्याकल्टी अपग्रेड गर्नका लागि हामी सधैँ लाग्न सकौँ र हामीलाई यो चिन्तन र दृष्टिकोण सधैँ प्राप्त होस् भन्दै मेरो दृष्टिकोणलाई यही बिट मार्न चाहन्छु। धन्यवाद। जय भारत।
Thank you, Sapna Bhatrai, for your wonderful Master of Ceremony. Thank you very much, Sapna. Chairperson of today's program, Anup sir, coordinator of Science Faculty, Chief Guest Principal of Balkumari College, Mr. Shiv Prasad Paudel, Vice Principal, Mr. Bharat Khana, management representative from teachers, Mr. Jagdishwar Khanal, coordinator of the education department, Mr. Ananda Kumara, all the faculty members of the science faculties, all the non-teaching faculty member of the science department, all the students. At the very outset, let me congratulate you all for organizing wonderful talk program on DNA. What to say about DNA? I'm not the student of the science and the pure science. I'm the student of the social science. So I don't know about the DNA. But what I know about the DNA is that DNA, the full form of the DNA, I think so, DNA is the divided with acid. I don't know. This acid basically is the heredity, I think, heredity materials in one and almost other organism of humans. So DNA types of DNA I know about DNA. One is the nuclear and another is a Yankee DNA. Mostly this type of DNA is available in almost all human body. All human body. So there are too many chemical base. I think so. There are the chemical base and there are four chemical base. ACGT. Therefore, a combination of this chemical base actually shows the building and growth of human body. I think so. So similarly, here when we talk about the DNA of the human body, similarly we have to talk about the nowadays the talk about DNA in social science as well. DNA of Balkumari College, the system of the pattern of developing the Balkumari College in a systematic manner or way, similar just like human body. Just human body, this is the system of the organism which is guided by the DNA. Similarly, Balkumari College, you are the product of Balkumari College. When you in the classroom or out the classroom, you have to show your DNA in the outside, positive DNA in outside the classroom. I would like to thank all the students, especially all the students of the Balkumari College Science Department for organizing such a wonderful program and supported by Lions Club of Balkumari College. And similarly, I would like to thank all the supporters, its own Biological Research Center and the Lions Club of Balkumari College for supporting our students in organizing this wonderful program. So it shows that Balkumari College does not do only greatest thing, but simple thing in greatest way. So that's why wish you a splendid and success of today's program. Thank you very much.
आफ्ना एबिलिटीहरू देखाउने मौका अवश्य दिनेछु। DNA धेरै नाम सुन्ने कुरा हो, स्पेसली हामी म्यानेजमेन्टका छात्रहरूको लागि यो पपुलर शब्द हो, तर हामीलाई धेरै अस्पष्ट शब्द पनि भइरहेको छ। कहीँ प्लेन एक्सिडेन्ट हुन्छ, मान्छेको भेरिफाई हुँदैन, तर DNA टेस्ट गर्नु पर्यो, बल्ल थाहा पाउन सकिन्छ। बिन लादेनलाई मारियो, यो हो कि होइन थाहा पाउनु पर्यो, ल DNA टेस्ट गरौँ। यानि अहिले के रहेछ भने एउटा जिन्स, यो मैले भन्नुभयो एउटा बुक्स, एउटा स्टोरी, जिन्स हो भने त्यसको लेटर DNA हो र DNAबाट बिस्तारै जिन्स निर्माण हुन्छ। यो तपाईँहरूको लागि त धेरै सुन्नुभएको कुराहरू होला, तर स्पेसल्ली यो साइन्स फ्याकल्टी अझ भने यो बायोलोजी फ्याकल्टी भन्दा बाहेकको लागि अझै पनि यो नयाँ शब्दहरू र यसका महत्त्वहरू के छन् भन्ने बारेमा तपाईँ भाइ बहिनीहरूले सरहरूले हामीलाई अझ जानकारी दिनुहुनेछ भन्ने मैले आशा लिएको छु। र मलाई के लाग्छ भने हामीले जे पढिरहेका छौँ क्लासमा, यसलाई व्यवहारमा विकसित गर्ने त घर परिवार समुदायलाई हामीले DNA भनेको के हो, यसको महत्त्व के छ, यसले दूरगामी असर के गर्नु छ हाम्रो जिन्समा, हाम्रो पुस्ताहरूमा, हाम्रा आउने भावी पिँढीहरूमा। आजकल DNA पुराना मैले एउटा फिल्म पनि हेरेको थिएँ, यो जुरासिक पार्क। एउटा DNA निकालेर अनि उसले नयाँ पुराना उसले डाइनोसरहरू बनायो। त्यो कथा लाग्छ हामीलाई, वास्तवमा म्यानेजमेन्टको कथा लाग्छ, तर मलाई लाग्छ साइन्सको विद्यार्थीको लागि कथा होइन, वास्तविकता होला हुनसक्छ। र यस कुरालाई हामीले बुझाउन सक्नुपर्ने हुन्छ, हामीले जानकारी दिन सक्नुपर्ने हुन्छ। हामी पनि जान्न चाहन्छौँ र मलाई लाग्छ बालकुमारीका विद्यार्थी भाइ बहिनीहरू धेरै जिनियस हुनुहुन्छ र आफ्ना प्रतिभालाई आगामी दिनमा अझ अगाडि देखाउन सक्नुहुनेछ र पछिल्ला आउने पुस्ताहरूलाई पनि तपाईँले सिकाउन सक्नुहुनेछ भन्दै आजको कार्यक्रम सफल रहोस् भन्दै म शुभकामना चाहन्छु। आज विश्व DNA डे उपलक्ष्यमा हाम्रो बालकुमारी कलेज साइन्स फ्याकल्टी जुन प्रोग्राम गरिरहेको छ त्यो प्रोग्राम बारेमा स्कुलमा मजा खुसी भइरहेको छु। हुन त अरूप पनि छ, यो एनर्जेटिक डाइनामिक सर हुनुहुन्छ। यस्ता प्रोग्रामहरू उहाँले ल्याउनुहुनेछ, मात्र उहाँलाई इन्करेज गर्नु पर्दछ र मलाई लाग्छ कलेजका प्रिन्सिपल सरले बेला बेलामा
Okay, please keep quiet. Respected chief guest, the principal of our college, Sibu Prasad Paudel sir, the vice principal Bharat Khanal sir, teacher representative of management committee Jagdishwar Khanal sir, President of Teacher Association, Mr. Ananta Dhumana sir, my senior faculty member, Dipendra Adhikari sir, Dr. Manoj Lal Das sir, Khadananda sir, Anil sir, Pavitra ma'am and Pradeep sir, and all my dear beloved students. First of all, today we are here for the World DNA Day, 25th April, and it's a small initiation that we are going to celebrate in this way, in this manner. DNA, deoxyribonucleic acid. What we are outside is reflected by the things which is inside. That is what we call the DNA. So my eyes are big, my face is bit larger. That is all the gift from my DNA, and I am happy for that. So we all are different with each other, and that's all because of the DNA. And most of the students, most of the people do not know about the DNA. So today's program is just the initiation to make you know much more about DNA. Nepal is a very beautiful country. It is gifted by the God. Various natural resources are here. But I think Nepal is very back in the field of the science and research. We have two great countries as our neighboring countries like India and China. India is doing great in the field of the science. They have launched Chandrayaan, that orbit the Mars and come back to there. But we are just the closest neighbor, and we haven't done anything in the field of the science. So why not to initiate? Nepal is also a developing country. We don't have so much resources. So even we cannot do the great research activities, then we can start the research. We can start something in the field of the science by doing such type of the activities. So Faculty of Science, with the help of two other organizations, initiate this program. And I would like to thank all the teachers, the principal sir, the management representative Jagdishwar sir, Bharat Khanal sir, Ananta sir, for being the part of the program and making the formal program very successful. Now, regarding the Balkumari College, Balkumari College is popularly known as one of the best college for the management. It has awarded best business college outside the valley. It's a proud feeling for us. But what I need now is that it should be considered as the best college for the science also. And that is possible only due to the combined effort of the principal sir with the faculty members and all the students over here. So, with this note, I want to add one thing more. Science is not easy. Science is not easy. It is an expensive program also. We do need lots of the instruments and equipments to do a small research. That is very expensive. And I know that the principal is helping us in this long run. We have recently installed the microbiology research lab here also. So I hope in future also the principal will provide his hand in so that we can uplift in the field of the science. So with this note, I would like to conclude my speech here, and I want to go to the next session, that is a talk program. So thank you, the MC, and thank you for all the teachers to be present over here, and thank you all. Jai Microbiology, Jai Environment, and Jai
Balkumari College Faculty of Science द्वारा आयोजित DNA संबंधित टॉक प्रोग्राम का सभापति साइंस फैकल्टी का प्रोग्राम इंचार्ज सर, हाई स्कूल प्रिन्सिपलज्यूहरू, व्यवस्थापन समितिमा प्रधान प्रतिनिधिज्यू, सदस्यका सभापतिज्यू, हाम्रो साइन्स फ्याकल्टीका प्राध्यापक मित्रहरू, कर्मचारी भाइ र सबै लेभलका मेरा प्यारा विद्यार्थी भाइ बहिनीहरू, अब मैले त अघि यो नजाने गाउँको बाटो सोध्नु भन्छन् नि, खान्छन् नेपाली। अहिले डिएनए साइन्सको टर्म अब हामीले के बोल्ने होला, यो त बरु हाम्रो विद्यार्थी भाइहरूलाई नि बोलाइदिनु पर्ने थियो कि जस्तो लाग्यो मलाई चाहिँ। विद्यार्थीबाट सुन्ने अनि साइन्स सरबाट सुनेर लास्टमा हाम्रो एक्सपर्ट अनन्त सरसँग सुन्न पाइन्छ भन्ने आशा लागेको थियो। यो त भाषण गर्न हामी पो आउनु पऱ्यो यहाँ है। अब तैपनि मलाई खुसी के अर्थमा लाग्यो भने हाम्रो भाइस प्रिन्सिपलज्यूहरू चाहिँ वास्तवमा एकदम विज्ञ भइसक्नु भएको छ। दुवैजनाले इक्जाम्पल सहित फुल फर्म सहित बखान गरिसक्नुभयो। वास्तवमा मलाई चाहिँ पटक्कै आएको थिएन, थाहा पाएँ अहिले, उहाँलाई धन्यवाद। अब वास्तवमा यो त के भन्दाखेरि हाम्रो पेपर प्रेजेन्टर जस्तै आजको चाहिँ मुख्य स्पिकर अन्नपूर्णी सर र अनिल श्रेष्ठ सर दुईजना, उहाँहरूबाट पक्कै पनि गहकिला कुराहरू सुन्न पाउनेछौँ हामीले। र हाम्रा विद्यार्थी भाइ बहिनीहरू पनि भविष्यमा सरहरू जस्तै वा सबभन्दा बढी यस सम्बन्धी असल वैज्ञानिक तपाईँहरू बन्नु हुनेछ भन्ने मेरो शुभकामना। र त्यसले भोलि देशको जुन क्षेत्रमा डिएनएको विज्ञताले देशलाई समाजलाई फाइदा गर्छ त्यहाँ तपाईँहरू पुग्नुहुनेछ, तपाईँहरूले देशलाई हेल्प गर्नुहुनेछ भन्ने पनि मैले विश्वास लिएको छु। र बालकुमारी कलेजको अघि नै कुरा आइसकेका छ, यो हाम्रो विद्यार्थी भाइ बहिनीहरू, पुराना भाइ बहिनीहरू, नयाँ र यहाँका फ्याकल्टीहरू सबै यस्तो प्रोग्राम गर्न अति जागरुक हुनुहुन्छ। वास्तवमा उहाँले हामीलाई भत्ता हुन्छ कि यस्तो प्रोग्राम गराउँछ भनेर। त्यस कारणले त्यो प्रोग्रामले एउटा कुरो त भर्खरै बिएससी पढ्न आउने भाइ बहिनीहरूलाई जानकारी हुन्छ भने हामी साइन्सको विद्यार्थी नभए पनि त्यसको बारेमा सामान्य जानकारी हामीलाई पनि हुने भयो। यसको लागि पनि म यो आयोजक लगायत सम्पूर्ण विद्यार्थी भाइ बहिनीहरूलाई धन्यवाद दिन चाहन्छु। र यो प्रोग्राम जुन मुख्य वक्ताहरू आउँदै हुनुहुन्छ उहाँहरूलाई सफलताको शुभकामना दिँदै यो प्रोग्राम राम्रो ढङ्गले सफल हुनेछ भन्ने विश्वास व्यक्त गर्दै मेरा केही शब्द टुङ्ग्याउँछु। हस्, धन्यवाद।
Actually, there are three types of the pyrimidine. To memorize it, you can memorize it as the CUT COT. That is the cytosine, uracil, and thymine. In case of the purine, there is what? Guanine and adenine. So, pyrimidine, that is CUT COT. I have already shown that in the previous slide. The next is nucleotide. So, nucleotide, I have already told you, it is the combination of what? It is a combination of the phosphate group, sugar, and the nitrogen base. If you go to the perfect structure, then look here. This is the phosphate, this is the deoxyribose sugar, and this is the nitrogen base, that is adenine. Okay? So this is the what? The nucleotide structure.
Okay, so that is a general introduction about the DNA. Now I think you are able to know what is DNA. So DNA is what? It is the nucleic acid that is made up of the what? Nucleotides. And nucleotides itself consists of what? Nitrogen base, phosphate and sugar. Now when we go through the structure, how the structure is arranged in DNA. Look there. There is a two strand of the DNA. If we put it in the linear form, then you can take it as one side this one, other side this one. So this is just like the ladder. Look here. This is the steps and this is the main pillar. And in pillar, what we have? Phosphate sulfur, phosphate sugar. Alternating molecule of the phosphate sugar forms the what? One portion of the ladder. The next backbone is what? Again the same. Alternating molecule of the sugar, phosphate. And the stem is made up of the what? Nitrogen base. A, T, G, C. And the bond in between the A and T is 2 hydrogen bond. In G and C, that is 3 hydrogen bond. But when it is in the natural form, then they are twisted helically like this. So this is the ladder form to make you easy. AT, GC, and this is the helically twisted form. Now after having the basic idea, then we should know what actually is the measurement or what actually is inside the DNA. Okay? So look here, DNA is a helix that is coiled twisted form which have the weight of what? 2 nanometer, that is 20 angstrom. And purine and pyrimidine base are stacked, means there is a purine and there is a pyrimidine phosphate. So sugar, nitrogen, phosphate forms nucleotide. Nucleotides forms the nucleic acid, and one of the nucleic acid is what? DNA. That is the general introduction about the what? DNA. Now what type of this sugar is present in DNA? There are 2 types of this sugar. One is called deoxyribose and next is called as the ribose. Now why it is called deoxyribose? Why it is called deoxyribose? Look at this carbon. Second carbon, there is no O over here. No O. Here it is. There is the presence of the O. So D means removal. Oxy means oxygen. Removal of the oxygen in second carbon. That's why the name is the deoxyribose. So the name of the DNA is from the sugar that is present in the DNA. Okay? Next. Sugar. Next is what? Nitrogen bases. So what are the nitrogen bases that is present in the DNA? They are adenine, thymine, guanine and cytosine. This is AG belongs to what? Purines, and TC belongs to what? Pyrimidines. Some will say be cool and be happy, always relax. In the exam time also, be relaxed, no tension, no worries. I answer, Goya answer. Okay, so this is the line, absolutely. This is the line. What will be the next step? If there is a student like you who is a bachelor, unmarried, then what is life? Life is to have a very beautiful wife. Let me say like that also. So, now what type of beautiful wife? Okay. So these all are what? Philosophical things. These all are philosophical things. The definition of life will change from one person to another. But in science, the definition will always be the fact. Everyone should say the same definition. Student, life is to study. Always. From morning to evening, you are just studying. So life is a study. And some student will say, sir, life is a struggle. I always have to struggle. I have to do my homework. I don't know about the chemistry. White man, he is having black babies. So he claimed that these are not my son and daughter. Okay? So he had done the DNA test in one program, and obviously these 2 children are not his children. DNA test is not positive there. Okay? So
this the miracle of the DNA is that paternity test can be 100% done from what? DNA. Okay. The next example is there. Look here. He is the program, what we call it, a VJ, like something like that. Okay. He used to conduct the program and he used to do the DNA test. So there is a black man, and he has 4 children, and he claims that none of these are his children. He suspects his wife, that means. So again, the DNA test has been done, and it was found that all these 4 are his children. Okay, so after DNA test, there is no any doubt, okay, for the paternity, for the parents. In Indian channel, there is always the huge news, बड़ी बहस, बात से लौकी गीता आखिर किसकी बेटी है? Okay, so Indian channel used to what? Given so many news, and they will put that in the highlight news. कसरी थाहा पाउने त हामीले है हामी एक आपसमा सबै फरक छौँ तर हाम्रो डिएनए कति मिल्छ 99% मिल्छ ओके 99% एक्ज्याक्टली हाम्रो डिएनए मिल्छ तैपनि यति धेरै भेराइटिजहरु किन because we have the sequence that we call it as the STR short tandem repeat polymorphism भन्छौँ हामी त्यसलाई है यसलाई म स्किप गर्छु ल्याङ्ग्वेजहरु धेरै छ है एउटा यो लोकाय हो लोकाय डु यु नो व्हाट इज दैट लोकाय मीन्स वन अफ द प्लेस जस्तै डीएनए को जीन हुन्छ जीनमा एउटा ठाउँ हुन्छ त्यो ठाउँ हो यो त्यो ठाउँको मैले स्टडी गर्दा एउटा रिपिट छ यहाँ GATA हामीसँग के हुन्छ नि डीएनए सिक्वेन्समा ATGC हुन्छ नि यस्तै यहाँ के भेटियो GATA भेटियो जस्तै एउटा एक्जाम्पल यसरी हुन्छ यहाँ कहाँ छ GATA खोज्नु पर्यो हामीले यहाँ छ GATA सो यो जुन सिक्वेन्स छ नि मेरोमा 20 वटा GATA छ भने अर्को मान्छेमा 12 वटा मात्र हुन सक्छ अर्को मान्छेमा 5 वटा हुन्छ सो दैट डिफर्स यति डिफरेन्सले गर्दा हामी यति धेरै फरक फेस भएका मान्छेहरु निस्केका छौं 99% DNA is almost just this difference makes us what differ from each other that is called as the short tandem repeats now just for example हेरौँ त यो मान्छेको AGAT 7 रिपिट छ AGAT यो सिक्वेन्समा 7 रिपिट यो सिक्वेन्समा 6 रिपिट छ यो मान्छेको 4 र 19 छ यसको सो यसले गर्दा यो डिफरेन्ट फेसेसहरु क्रिएट हुन्छ यो डिफरेन्ट फेसेसहरु त्यसले गर्दा क्रिएट हुने हो ओके जस्ट अब वन एक्जाम्पल हामी लियौं just for fun compare the profiles of 3 men with that of a mother and child to determine the biological father यहाँ मैले एकछिन होला यो मदर छ मदरको मैले डिएनए लाई टुक्रा पार्न मिल्छ है सेकेन्ड इयरको लागि थाहा हुन्छ रेस्ट्रिक्सन इन्डोन्युक्लियस इन्जाइम लगाएर र त्यसलाई मैले रन गरे डिएनए को टुक्रा यसरी मैले रन गरे जेलमा भने एउटा फोटोग्राफी निस्किन्छ त्यसबाट ओके सो मदरको यो भयो चाइल्डको यो भयो अब यो तीन जना ड्याड बायोलोजिकल ड्याड को हो त तीन जनाले क्लेम गर्यो Now just guess, dad 1, dad 2, dad 3. Who is the biological dad? Dad 2. Look here, your answer is dad 3. Look here. यो पहिलो जिन हेर्ने हामीले चाइल्डको पहिलो जिन हेरौँ यो पहिलो जिन कसकोमा छ मदरको छ छैन पक्कै फादरबाट आयो फादरमा हेर योसँग छ पहिलो जिनले 11 ड्याड भने 11 ड्याड भने सेकेन्ड जिन हेरौँ छैन मदरसँग छ यसको थर्ड जिन हेरौँ मदरसँग छ फोर्थ जिन हेरौँ योसँग छ त छैन यो ड्याड हैन एउटा मात्र मिल्यो अघि यो मिलेन त कोसँग मिल्यो दिस सेकेन्ड पनि योसँग मिल्यो अर्को लास्ट योसँग भएको जिन कि मदरसँग हुनु पर्यो कि फादरसँग हुनु पर्यो त्यसपछि मात्र दिस इज द कन्फर्म दैट थर्ड इज द बायोलॉजिकल ओके सो दैट इज द मिरेकल अफ द डीएनए ले यस्तो अचम्म गर्छ नेक्स्ट डीएनए बाट हामीले के पनि गर्न सक्छौं क्रिमिनल इन्भेस्टिगेसन अनि सीआईडी भन्छौँ हामी हैन सो क्रिमिनल इन्भेस्टिगेसनमा डीएनए ले धेरै रोल खेल्छ जस्तै फर एक्जाम्पल लुक हियर डीएनए डजन्ट lie नेभर lie है डिएनए टेस्ट एनालाइसिस एन्ड इम्पोर्टेन्स मान्छेले पहिला रौँ ब्लडका थोपाहरू यिनीहरू भयो भने त्यो चाहिँ स्याम्पलको रूपमा लिन्थे अहिले त्यो मात्र होइन च्विङ्गम लिपस्टिकका दागहरू होइन रौँ है अनि यो ह्यान्डको छापहरू ह्यान्ड बनाऊ र सस्पेक्टलाई पत्ता लगाऊ होइन फेरि एउटा इक्जाम्पल हेर्नु त जस्ट फर इक्जाम्पल देयर इज अ कम्प्युटर डिएनए कलेक्टेड क्राइम सिन त्यो त्यहाँ क्राइम सिनमा चक्कुले भिक्टिम चाहिँ त्यो सस्पेक्ट 1 2 3 छ है एकदम एकदम सरले भन्नुभएको छ मस्किटो यो क्राइम सिनमा यहाँ ब्लड लाग्छ ब्लडमा भएको डिएनए चाहिँ डाइरेक्ट लाग्छ सस्पेक्ट के हो त DNA is the confirmed test. That is the next miracle of the DNA. Now, the next. Allah says there is nothing right. Okay, the next is that the first bioluminescent plant. Bioluminescent भनेको जस्तै luciferase enzyme भनेर हामीले जुनकिरीको थाहा छ जुनकिरीले राति बत्ती बालेर हिँड्छ हैन अर्को बत्ती बाल्ने भनेको ब्याक्टेरियाहरूको जिनमा बायोलुमिनेसेन्ट जिनहरू हुन्छ जसले गर्दा ब्याक्टेरियाहरू बल्छ त्यो जिनलाई डिएनए मा हुन्छ जिन that gene has been taken out from the bacteria and incorporated into the plant. So this is not the imaginary picture, this is the real picture. Okay, plant ma ke halyo bioluminescent gene halyo plant lai ke garyo balyo. अब you don't need to light the street lamp. Street मा के लगाउने रुख लगाऊ है रुख आफै बल्छ राति के हुन्छ उज्यालो हुन्छ र हाम्रो देशको लागि के रे झन् फाइदा छ नेपाललाई धेरै फाइदा छैन फाइदा छ आफ्नो बसेको रुममा एउटा गमलामा एउटा प्लान्ट हालौँ राति बल्छ लोड सेडिङ भयो भने नो प्रब्लम त्यो बल्छ भइहाल्छ हैन Next is look here. DNA next miracle look here. This is what? This is the chicken. These all are chicken. Featherless chicken. Featherless chicken. Right? Boiler chicken are meat for the meat only. मासु खानालाई हैन त चिकन अब त्यो जनियो पकेटा सकेटा यो के फेदर निकाल्नलाई जाऊ भएन पानीमा छु के गर्दियो फेदरको जिन नै हटाइदियो फेदर निकाल्ने जिन नै अमिट गर्दियो अब के छ त featherless chicken कल्पना हैन दिस इज द रियालिटी right European countries ma featherless and I have already told you once I am also suffered from the what galba एकदम fever high fever flu like fever हैन break bone fever भन्छ एकदम ढाडसाड दुख्ने एकदम बोनहरु भित्रैबाट पेन हुन्छ है सो त्यो डेंगु केले गर्दा हुन्छ मस्किटोले गर्दा हुन्छ अब मस्किटोको जिनमा डिएनए मा केही चेन्जेस ल्याइदियो ओके के चेन्जेस ल्याइदियो भने यिनीहरुको जिनमा एउटा लेथल जिन भन्ने हाल्दियो मेलमा त्यो मेललाई चाहिँ यिनीहरुले कल्टिभेट गर्यो र मार्केटमा भनौं न पठाइदियो इन्भाइरोमेन्टमा अब यिनीहरुले मेटिङ गर्दा लुक्यो यो मेलसँग मिटिङ गर्दाखेरि यसले जुन प्रोडक्ट दियो नि यो पछि डेभलपै हुन सक्दैन किनभने त्यो लेथल जिनले त्यो बच्चालाई के गर्छ मार्छ उ एडल्टमा कन्भर्ट हुनै सक्दैन के एडल्टमा जानु भन्दा अगाडि नै मर्यो सो मस्किटोबाट लाग्ने रोगहरू मलेरिया डेंगु जापानिज एन्सेफलाइटिस all will be solved due to the what certain dead mosquitoes that is genetically modified organism right यो चाहिँ कुन कम्पनीले गर्यो ब्रिटिशको बायोटेक कम्पनी कल अक्सिटेकले गरेछ एडिस इजिप्टी मस्किटोमा है त्यति धेरै कामहरु भइरहेछ डिएनए मा नेक्स्ट इज लुक हियर एफआईवी डु यु नो एचआईवी एचआईवी भनेको ह्युमन इम्युनोडेफिसिएन्सी भाइरस हो त्यो ह्युमनलाई मात्र लाग्छ अब यहाँ फेलाइन इम्युनोडेफिसिएन्सी भाइरस हो जुन क्याटलाई मात्र लाग्छ क्याटमा पनि के छ इम्युनोडेफिसिएन्सी भाइरस छ क्याट फ्यामिलीलाई पनि के हुन्छ रे एचआईवी क्याटलाई सर्छ सर्दैन नाम नै के हो रे ह्युमन इम्युनोडेफिसिएन्सी भाइरस हो र भाइरस कहिले पनि एकजनालाई लागेको भाइरस अर्कोलाई भनौँ न एउटा स्पिसिसलाई लागु भाइरस अर्कोलाई सर्दैन। जस्तो फर इक्जाम्पल मैले भन्थेँ हामीलाई बर्ड फ्लू लाग्छ भन्थ्यौँ हामी होइन, बर्ड फ्लू हामीलाई लाग्छ, नामै के हो बर्ड, हामी बर्ड हौँ। तर बर्ड फ्लू हामीलाई किन लाग्छ, किन रिस्की छ भने त्यो बर्ड फ्लू हाम्रो शरीरमा पसिसकेपछि त्यो केमा कन्भर्ट हुनसक्छ, ह्युमनमा कन्भर्ट हुनसक्छ, त्यसले चान्स छ। सो बर्ड फ्लू जाँदैमा हामीलाई रोग लाग्छ भन्ने छैन, तर त्यो म्युटेट हुने अवस्थामा पुग्यो भने त्यो म्युटेट भएर ह्युमनलाई के गर्छ, रोग लगाउँछ। बर्ड फ्लूका भाइरसले हामीलाई रोग लगाउने होइन। ओके, त्यस्तै गरी एचआइभीले काउलाई गोठलाई कहिले पनि इम्युनिटी लाउँदैन, उनीहरूको आफ्नै भाइरस छ एफआइभी। र यसको स्टडी गर्ने क्रममा यो क्याटको स्टडी गर्ने क्रममा चाहिँ के गरिदियो, यसमा लुमिसिन हाल्ने। हेर भाइ, इङ्ग्लिस मिडियम कुहिरेहरूले यत्तिकै मुभी बनाउँदैन, दे आर द साइन्टिस्ट, साइन्स फ्याक्ट कुरो मात्र बनाउने हो। हाम्रो जस्तो मुभी बन्छ र त्यहाँ गीत गाउने पाहाडमा नाच्ने यति उति होइन, त्यस्तो होइन। दे टक अबाउट द फ्याक्ट्स। स्पाइडरम्यान यत्तिकै बनेको मुभी होइन, स्पाइडरम्यानलाई धेरै सोचेर बनाएको हो। स्पाइडरम्यान साँच्चीकै रियल लाइफमा पनि के छ, पोसिबल छ है। तर अब मुभीमा त अलिकति देखाउनु पऱ्यो, त्यो यसो पठाएर झुन्डेर यताउता जान्छ, त्यो चाहिँ होइन, त्यस्तो चाहिँ हुँदैन। तर मान्छेको हातबाट हामीले स्पाइडरको जाली निकाल्न मिल्यो। ल अब यहाँ हेर, जस्तै स्पाइडर बोट्समा हेर त, यसमा चाहिँ बोटमा स्पाइडरको जिन हाल्यो, स्पाइडरको कुन जिन हाल्यो, जाली निकाल्ने जिन। यो जुन जाली निकाल्छ त्यो जाली एकदम स्ट्रङ हुन्छ, त्यसलाई चाहिँ जुन प्यारासुटमा हालिन्छ नि डोरीहरू, यिनीहरू चाहिँ सिल्कै भनिन्छ है। अ गोट द्याट प्रोड्युस स्पाइडर वेब प्रोटिन इन इट्स मिल्क। गोटमा त्यो जिन हाल्दा गोटको मिल्कले के प्रोभाइड गर्यो, जाली प्रोभाइड गर्यो, जुन चाहिँ सिल्क मिल्क भनेर भन्छौँ हामी है। त्यो धेरै कुरामा हामीले युज गर्न सक्छौँ। फर इक्जाम्पल आजकल के भइसक्यो भने साइन्सले धेरै प्रगति गरिसक्यो। इन केस अफ स्टेरिलिटी अल्सो वी हेभ अ टेक्निक व्हाट वी कल इट एज अ टेस्ट ट्यूब बेबी। इन विट्रो फर्टिलाइजेसन गर्छौँ, स्पर्म्स ओभालाई ल्याबमा मिक्स गरेर हामी के गर्छौँ, इम्ब्रायो डेभलप गरेर फेरि मदर युटेरसमा हालेर बच्चा जन्माउने। यो त थाहा छ नि, नेपालमा पनि यो सुविधा छ होइन। अब यहाँ के भयो भने कसैको मिल्कमा चाहिँ टोटल एमाउन्ट हुने यसो थाहा छ, यो फ्याक्टर 8 जुन छ यो यसमा छैन भने यसलाई हामी पहिला भाइरसमा हाल्छौँ, भाइरसले ह्युमनलाई इन्फेक्ट गर्छ, इन्फेक्ट गरेर ह्युमनमा त्यो डिएनए न्युक्लियसमा हालिदिन्छ र हाम्रो न्युक्लियसमा यो डिएनए गएर हाम्रो न्युक्लियसमा बसिसक्छ। हाम्रो न्युक्लियसले के निकाल्छ त, यो प्रोटिन निकाल्न सक्छ। तर हामीलाई कसको आवश्यक पर्यो यहाँ, भाइरसको है। भाइरस एज अ भेक्टर है। अब यो डिटेलमा हामी पछि फेरि कुनै दिन बुझौँ है त। सो जिन थेरापी भयो। अर्को छ डग पोपी पिग्स है। यहाँ के भन्छ, पिग्स द्याट आर बोथ मिट एन्ड भेजिटेबल। साग खानलाई गाह्रो भयो हामीलाई, साग हाम्रो मन परेन, अब पिगमा सागको जिन हालिदिने, साग खाए बराबर है। वेस्टर्न कन्ट्रिजहरूमा सागहरू अलिक कम पाउँछ क्या है, ग्रिन भेजिटेबल्सहरू कम पाउने भएकोले यिनीहरूले के गरिदियो भने पोपी पिग्समा चाहिँ त्यो जिन हालिदियो। त्यो जिन हालिसकेपछि एउटा प्रब्लम के भयो भने सेचुरेटेड फ्याटी एसिडहरू अनसेचुरेटेडमा कन्भर्ट है। एक्वेरियमलाई एकदम राम्रो बनाउनु पर्यो। के गर्नु, हामी कलरफुल फिसेसहरू लाउँ, कलर्स इन फिसमा हाल्यो भने वी हेभ द व्हाट, दिस कलरफुल फिसेस। ओके, सो विथ दिस रिगार्ड लास्ट वान, यो जुन व्हाइट छ यो व्हाइट के हो, राइस हो। दिस इज कल्ड एज द गोल्डन राइस। गोल्डन राइस भनेको सुनको जलप भएको हो होइन। यसमा क्यारोटको जिन हालेको, क्यारोटको जिन अहिले 80 भर्खरै पैसा कति गर्दै भिटामिन ए क्याप्सुल फ्रीमा के गरेको थियो ल्याएको थियो होइन, कति कति हो। सो अब भिटामिन ए क्याप्सुल बच्चालाई खुवाइराख्नुपर्छ भने जरुरत नै भएन। यदि त्यो बच्चालाई कुन चामल खुवाइयो भने, गोल्डन राइस खुवाइयो भने, गोल्डन राइस आफैमा के छ, भिटामिन ए छ है। गाजर मेवा आँप भनेर खोजी खुवाइराख्नु परेन। द्याट इज अल्सो द मिराकल अफ द वर्ल्ड, डिएनए। र यी सबै कुराहरू बाहिर कन्ट्रिजमा आइसक्यो, तर नेपालमा चाहिँ है। So the power of DNA is मैले भनेरै भ्याउँदिनँ adds for color, texture, rhythm and blah blah everything to your life. Exit. Hold on, silence. Hey, hold on.
Talking about the polymerase chain reaction. So many of these students knows about it. So the polymerase chain reaction is the reaction in which we can amplify the DNA. So you have known about the DNA. So whenever there is any criminal activities, there will be only less amount of the sample. For example, the amount of the blood will be very small. So to perform the DNA fingerprinting or the gel electrophoresis, such type of the DNA has to be amplified. It means the smaller amount of the DNA cannot detect whether the suspected one is the criminal or not. So in that case, we have to amplify the DNA. So that is called the polymerase chain reactions. And one of the instruments used for the polymerase chain reaction is the thermocycler. So we will discuss about it. So this instrument is available in Nepal too. So all these students who are going to do the thesis can use this type of instrument in order to detect the particular gene, because this is also used for detections of the particular gene. So I have seen some of my students are working on the ESBL, that is the extended spectrum beta-lactamase, one of the positive gene for the antibiotic resistance, and that can be detected from the PCR, which is called the polymerase chain reaction. One of my students is working on the methicillin resistant staphylococcus, which are being isolated from the NOTs or the health workers. So that methicillin resistant staphylococcus aureus can also be genotypically confirmed. That can be genotypically confirmed. We can confirm phenotypically by the means of the antibiotic susceptibility test, but genotypically you can confirm by the use of the PCR. So what is the PCR? This is a technique in the molecular biology that uses each stable polymerase to amplify a particular DNA sequences as specified by the primers. So you must be new with some of the terms like the polymerase and the primers. So these are the new terms for you. So this polymerase is the enzymes. So you have known about the central dogma of life. The central dogma of life. In the central dogma of life, there is the replication. One part is the replication. So replication stands for the amplifications of the DNA. So that is inside the body. So replication happens inside the body, but this PCR happens outside the body in the instrument. So if you see, so for that we need the polymerase enzymes. So the polymerase enzyme is the heat stable polymerase enzymes. So our body enzymes can tolerate up to 60 degrees centigrade. Our body enzymes can tolerate up to 60 degrees centigrade. But if you see here, so how much heat we need is that we need about 94 degrees centigrade heat. Our body can tolerate the 60 degrees centigrade of the heat, but we have to we have to give the 94 degrees centigrade heat in order to denaturate the DNA. So I come here. So next is the primer. So primer are the short segments of the DNA that is used in the polymerase chain reactions. Using a PCR in the short time procedure, produce the millions copies of the targeted DNA sequence. So from the one copy of the DNA, so millions copies of the DNA can be synthesized. So PCR uses thermocycling, which consists of heating and cooling of the PCR cycle to a defined series of individual acid amplifications of the PCR sequence. That means by using a specific primer, you can amplify a specific gene. Most PCR methods dynamically amplify the DNA fragments of between 0.1 to 10 kilobase per year. Although some techniques allow the amplifications of fragments up to the 15 kilobase per in size, the PCR was developed by the Carly Mullis in So the techniques has revolutionized the medical and the biological research. There's a variety of applications. So it can be used for ranging from DNA cloning to the disease diagnosis. So in 1993, Mullis was awarded Nobel Prize in Chemistry for his contribution to such a prolific techniques. So what is the principle of the PCR? So the principle of PCR has the 3 steps. So one is the denaturation step, next is the annealing step, and the last one is the elongation of the extension step. So these 3 steps are there in the PCR. Other principles of the PCR. So under the consists of the various things. For example, so you are working on the antibiotic resistance and you have to find the make agent. For example, only this segment is the make agent. So you have to amplify the make agent. In order to amplify, first we have to denaturate the DNA, then after we have to use the primer, and there will be the annealing steps. The annealing steps is the additions of the primer to the template DNA. The template DNA, in the template DNA, the primer will add. Then after there is the next step called the elongation step, in which the DNA will be amplified and the nucleotides will be added in the 3' end of the primer. So this is what the principle of the PCR is. So you can see this DNA. So if you see, this is the double stranded DNA. So this double stranded DNA is converted into the single stranded DNA by the applications of the heat. By the applications of the heat, this double stranded DNA can be converted into the single stranded DNA. So now is DNA, look at this DNA, this acts as the template for the new DNA synthesis, and this also acts as the template for the new DNA synthesis. So this will synthesize the mucin, and for the synthesis of the mucin, we require the various enzymes. As I have already told you, one of the heat stable polymerase gene is required. Okay? So our body cannot go beyond 37 degrees centigrade, because whenever our body reaches to 60 degrees centigrade, even our enzymes are denatured. But you can see in the thermocycler, our temperature will reach up to the 94 to 98 degrees centigrade. Remember this point. In the thermocycler, the temperature will reach to the 94 to 98 degrees centigrade. In such system, we will imply that cannot be replicated or cannot be polymerized in the thermocycler by our own polymerase enzymes, because our polymerase enzymes is unable to tolerate this temperature. So to overcome this problem, scientists have isolated the polymerase enzymes from the thermophilic bacteria known as the Thermus aquaticus. So one of the thermophilic bacteria known as the Thermus aquaticus. So the bacteria has the enzyme, polymerase enzyme, that can tolerate the high temperature. So we use the polymerase enzymes of the Thermus aquaticus. So the first step is the denaturation steps. So typically it consists of the 3 steps. One is the denaturation, in which the double stranded DNA are changed into the single stranded. Next is the annealing steps. In these steps, the primer gets joined or add to the single stranded DNA. Next is the elongation steps, that is done by the DNA polymerase enzyme. So these are the steps. And if you see the instrument that we use for performing the polymerase chain reaction is the thermocycler. The word thermocycler is given because this goes into the repeated cycle. This temperature goes in the repeated cycle. So this is called the thermocycler. The machine is called the thermocycler. So, and this is a very expensive instrument, because within a second, the temperature from the 98 can be reached to the 60 degrees centigrade. So this instrument is very much expensive because no any instrument is there that can lower the temperature from 94 to from 98 to 60. There is not an instrument, so this is only the instrument that can lower the temperature. So the first step of the regular cycle consists of the heating of the reaction to 94 to 98 degrees centigrade. So what we do, we heat the DNA to 94 to 98 degrees centigrade. Double stranded DNA is denatured, yielding a single stranded DNA molecule. So you can see the double stranded DNA are converted into the single stranded DNA in the topic called the denaturation. The temperature employed in this step is the 94 to 98 degrees centigrade. So next is the annealing steps. So in the annealing step, what we do, we lower down the temperature in about 50 to 65 degrees centigrade. So at first we rise the temperature, then after the conversion of the double stranded DNA into the single stranded DNA, we put the temperature to the 50 to 65 degrees centigrade, and the primers are added to the elongation step. So in the elongation step, the work is done by the polymerase. So we have written here Taq polymerase. So the Taq polymerase means T stands for the thermos, AP stands for the aquaticus. So it is the polymerase enzymes of the thermos aquaticus bacteria. So what happens is that, so DNA strands will break down. If you remember noise here, right? So I will run it fast. Okay, so what is the uses of the PCR? So what you can do with the help of the PCR? As this instrument is available in Nepal, and even a little technique, you can use this machine. So what may be the method, what may be the uses? So selective DNA isolation for other processes such as the Southern blotting, so Southern blotting, sequencing, inertiation into the plasmid vector, etc. So you have heard about the DNA fingerprinting. The DNA fingerprinting is done by the method called the Southern blot. So you can do by this Southern blot that was discovered by the scientists named as the Southern. So this test is given the Southern. So next is the sequencing. So there are various methods for the sequencing. One is the Sanger method, next is the automated sequencing method, next is the next generation sequencing method. So insertion into the plasmid vector is one also uses of the PCR. So you can also detect the mutations. So in the bacteria, if there is the mutation, you can detect the mutations by the help of the PCR. Similarly, it helps in the amplifications and the quantifications of disease diagnosis, estimations of the viral loads, and DNA fingerprinting, for example, to preserve the parental disputes and the criminal side investigation. So these are some of the uses of the PCR. So next uses. So I have seen some of my students working in the MRSA. So I have already told you we can amplify a shortened sequence of the DNA by the PCR machine. For example, you are working on the methicillin basis of the magazine. So suppose the size of the magazine is 533. So what you can do, you can use the markers. In the lab A, we have used the marker. This is the marker, and in the lab now we have also used the marker. So the marker is like the scale. So it helps to detect the size of the DNA. So you can see here 100, 200, 300. This is the size of the DNA. And what is the size of the magazine? The size of the magazine is about 533 base pairs. So after you amplify the DNA, so what happens is that only the magazine will be amplified. Other things won't be amplified. Only the magazine will be. The presence of the magazine, no any bands will be obtained like this. So 2 represents the no any presence of the magazine. If there is the magazine, what happens is that you can see in the 500, so near to the 500 there is the next one. You can see this is the DNA of the bacteria. So we have amplified the magazine. So in the land 3 we have put this amplified DNA. So this DNA, the size of the DNA is nearly equal to the 500, and the magazine is about the 533. From this information we can find that the bacteria has the magazine. So we can get it from this one. So So next is you have already known about this one. So all in earlier presentations you know about the criminal investigations. So that can be performed from the southern plotting. So next there was the mini satellites, there was the BNTR, there was the STR. So these are some of the terms that is used in the this DNA fingerprinting. So what happens is that in the DNA, in each one of the DNA in the humans, there is one region called the telomerase region. The telomerase region is not good for any of the amino acids. These are not responsible for the transfer of the any character in the humans or any of the animals. So the work of the telomerase defines the lifespan of the humans. So if you have the larger number of the telomerase, so the life of the humans is greater. So this telomerase can also be used for detecting whether that is the criminal, whether that is the parents or not, or in the DNA fingerprinting, we can use that telomerase region. So what happens is that in the telomerase region, there is the repeated sequences known as the variable number tandem repeats. And this variable number tandem repeats can be, cut by the help of the respiration enzymes, and that can be put in the gel electrophoresis. After you put in the gel electrophoresis, this has to be plotted on the nitrocellulose paper. We take out the nitrocellulose paper and we add the, we add the DNA props. And after we add the DNA props, the bands can be seen in the UV, under the UV radiation. So this is how we can do. So this is already discussed over. Okay, this is what I am talking about today. Thank you for your time.
Okay, thank you very much, Anil sir, for giving. Okay, with this note, thank you very much for your participation. So today on DNA Day, we come to know something about the DNA, and I request you all to have much more information regarding the DNA through the literature review also. Okay, we being science students have to do lots in the field of the science. Thank you for your participation. Thank you all.